Sure. Yeah, so thanks Bronson, and thanks for the invitation to talk today. My name's Kyra. I'm going to show you some work I did up until the beginning of this year in Princeton University and now at the University of Melbourne, where I am in the Gino and the Smith group. So, because I guess we're all interested in organic semiconductors, we probably know a bit about organic photovoltaics and how they work and how you'd want to take energy of photons and change them into charges that you can use. In the work that I've been doing for a while now and recently, we're really interested in getting to see in detail all of these different processes from photon absorption through to charge migration. And we can do that with these ultra-fast methods and really track in detail what happens in each step of the process. But organic photovoltaics are not so simple in their structure. So we know now that nanoscale effects have really large impact on efficiencies of all different types of devices. But particularly in organic photovoltaics, you might have in the early days this sort of really simple bilayer where you have your donor acceptor materials. But now we know that the device structure that works much better is this really complicated nanoscale interpenetrating network of donor and acceptor. So, I mean, this sort of structure was formed to help excitons diffuse to the interface, have a really large interfacial area, but really the nanoscale morphology is affecting the efficiency of every single step along the way. I'll say now with sort of efficient devices in mind, now we're up to sort of 17, almost 18% efficient devices. It's been shown that all of these steps we're kind of getting to a point where they can be really quite efficient. And it's been noted by quite a number of people that it's really the charge migration, the recombination step at the end here that really has the largest impact of efficiency in the best devices that are out now. So with this in mind, we have free carriers, electrons and holes. We want to harvest them. We don't want them to recombine, but they're mutually attracted to each other because they're positive and negative charge. So to put this into some context, in other light harvesting systems, so we have existing photovoltaics where you might have a silicon crystalline lattice. You're also obviously trying to harvest free charges. You have a really simple morphology. You really have no nanoscale complexity to these devices at all. The reason you can harvest free charges is because you have things like high dielectric constants and really good screening, low exciton binding energies, but you really have no complexity there. This is interestingly in contrast to what's going on in, oh, can you guys see? I just want to check you can see the next slide, or am I?
We're looking at your title slide at the moment.
Oh no, okay. I'm like through to slide 3, so I'm not sure what's going on, but I was just talking for a long time. So when I change, it's not, you're not getting it?
No, so at the moment we just see your title slide.
Huh. All right, it's telling me that sharing is paused. I don't know what it is. Now it tells me I'm screen sharing again. Can you see anything now?
Yeah, so now we're seeing your like where you're editing your slides.
Huh. All right, sorry, just give me 1 second. Anything now?
Yeah, so harvesting charges.
Okay, good. And if I change, you're seeing that?
I still see harvesting charges.
Wow, this is so weird. Has anyone had this problem before?
No, I've never seen this either. I mean, it seems to work when it's not full screen. So maybe the workaround for now,
yes,
if you don't mind, is just, yeah, the sort of this view.
Okay. Secret slides.
Yeah, like it's kind of a not ideal, but it seems like this will do the job.
Okay, so how about this?
Yeah, I think that should get us going. Are you able to advance?
Are you able to see this?
Yes. I think, yeah, this is a bit awkward, but this will have to do.
Okay, yeah, I'm sorry. I really apologize for that. I'm not sure what's going on, but okay, so where we were was just talking about, I guess, yeah, I had some images up, so talking about tracking these processes in charge photogeneration, how the morphology matters of the bulk heterojunction like this, and then going through to understanding a little bit about how we harvest light in existing PV, and then how we, really how nature does it. So the contrast here is really that in photosynthetic reaction centers where positive and negative charges are separated, we have really a really complicated nanoscale structure of chromophores arranged in this particular way. And you actually get an energetic cascade that's able to spatially separate mutually attracted charges. And it does this with almost a perfect quantum efficiency. So this real complexity is a good thing in this system, and it's really kind of amazing how this works. So I guess now we're in this regime of new technologies that aren't perfectly crystalline in these, perfect lattices. We have all this complexity. And so the question is, do we view this as something that's a challenge, or is it maybe an opportunity? Okay, so what I'm going to talk about today is looking at these actions of the bulk heterojunction and seeing what happens as you go from photons to charges that are harvested, seeing all these steps. But what we found is that there's another function to the bulk heterojunction. And that's that actually the morphology can play this role of accumulating charges on the nanoscale. And what we see, really what we didn't expect, is electric fields, really tiny but very large electric fields building up between domains, much like you'd see in a capacitor. Okay, so to get to introducing the specifics of this work, we were looking at this material blend of a donor material called BQR from the group of Dr. David Jones at University of Melbourne. And it's really interesting and nice material, and blending that with electron acceptor PC71BM. So you can do this, you're harvesting a bunch of the visible spectrum. And then when we blend these materials together in a sort of one-to-one blend, turns out you can make a device that's around 5% efficiency. This is just a cartoon of what these donor acceptor molecules look like in their mostly intermixed arrangement. And when we have some sort of annealing treatment that happens to this film, so here it's a really gentle treatment called solvent vapor annealing. You expose this whole film to a solvent vapor, and here it's THF for only 20 seconds. The vapor swells the matrix, it swells the film, and it's able to make these really small crystallites via Oswald ripening of the BQR molecules, and they're around 30 nanometers in size. So a little bit of crystallinity, but the effect that this has is by effectively doubling the device efficiency. So we're interested in why this makes such a big impact on device performance, and we're interested in studying this in really a lot of detail, how these ultra-fast events unfold, and these details in between photon absorption and charge migration. And we do this with transient absorption spectroscopy, which in short, instead of taking a ground state UV-vis or absorption spectra, we're first pumping and then taking really an absorption spectra of excited states and tracking them in time by using really short pulses. Okay, so another thing I need to briefly go through is to talk about the Stark effect. So this is a separate type effect to just regular absorption, where I talked before about electric fields are involved, and this is what how electric fields would impact your chromophores absorption emission of light. So it's also known as electroabsorption. So say we have some absorbing chromophore here, like in green, it has a transition dipole moment. If you turn on some electric field across this molecule, what can happen is that can change the energy of these states, and it can change the absorption of this. And this can be to do with the dipole moment or the polarizability, but if it's affecting the dipole moment, it turns out the orientation, or relative orientation between this electric field and the transition dipole moment really has the big effect on which way the energy levels get shifted. Now here I've shown some sort of voltage that we've on purpose put across this molecule, but you can have photo-generated charges also causing this effect. So here's an example. You might have an electron in a hole with a field between them, and the field and the relative orientation of that dipole are set. And so you'd see a shift in the electric, in the absorption spectra in one direction, from without electric field to with. If you take the difference of these, what you end up with is the first derivative of the absorption spectra you started with. If you have the case in which there's a lot of disorder and a lot of different mutual orientations between the transition dipole moment and the electric field, you actually get a whole bunch of different things happening, shifting higher and lower in energy. And the difference between this is always going to end up being a second derivative line shape. So if you're kind of interested in these effects and the Stark effect, what you would generally do is take your absorption spectra and just take the first derivative and the second derivative of that line shape. And then you're going to get a good idea as to what these different Stark effects would look like, which can be really useful. Okay, so to keep going and look at some spectroscopy, what I'll show is first we just looked at this donor material anyway. That's what we were exciting. And this is in a film. And so we can look, here we're going up in time on a log scale in picoseconds. And straight after photoexcitation, we see a signature of an exciton. Because there's nowhere for the exciton to go, no charge transfer is going to occur. We just have the lifetime of that exciton decaying back to the ground state here. That's really all that's going on in this film. Now, when we add our acceptor material, our PC71BM, we have excitons, and now we have a new species that we're making almost straight away, which is our charges, our whole polaron here that we're able to detect. And here's some slices through showing this evolving in time. I'll then show, so this first panel is showing what happens when you have this AUCAOS device with about 5% PCE. And then after our really subtle annealing treatment, what you can see is we also have excitons turning into charges and almost in an identical type way. Then it's sort of our job to spot the difference and see, well, there has to be something different with these two different samples. And indeed there is. You can see this over here around 700 nanometers. There's a new signal growing in. You can see this even better maybe with the line shapes in our slices over on the right. So what we see is we have this really sharp kind of feature compared to everything else, and it's growing in with time, whereas everything else is decaying. Okay, so these sort of sharp, sort of derivative type signals kind of reminded us already of this Stark effect that I just mentioned. And here I've showed overlaid the second derivative of our absorption spectra with the BQI exciton we know is in here as well. A few species that are overlapping. And if you look at the line shape here, you see really it's overlapping with stuff for sure, but it really looks similar to what's going on. So we believe we do have an exciton, a polaron, and then this electroabsorption signal all sort of up in here as well. Okay, so because these signals are all overlapping, we looked in more detail to pull them apart. We know we have our exciton species, our polaron, and then here's what's left over in blue, this electroabsorption EA signal. We track them in time then. Now we have the species to pull out. We have our exciton decaying, our polaron that's there at the beginning, but it builds up a little bit, probably due to exciton diffusion. And then after that polaron, those charges have been there for some time, our electroabsorption is building and building over time and holding for the length of our experiment, so several nanoseconds. So we can confirm that this line shape is what we think it is. We're comparing with the second derivative. We also got our colleagues, Princeton in the engineering department, to make us some devices and crank up really large voltages across molecules of BQR. We saw this response in the absorption. So really, here is shown in gray. It really does match up really nicely with what we've pulled out from our transient measurements, where we don't have any electrodes, we just have the device active layer and photo-induced charges being formed. We also did some further experiments to confirm that all the dynamics are power dependent. We really think that this electroabsorption is due to free carriers rather than bound geminate carriers in this device. And the handy thing about doing these device measurements is we've got a calibration factor. So we knew how much field you had to put into this material to get this type of absorption response. This scales quadratically, and we're able to find that we almost have half a megavolt per centimeter square of electric fields. So really pretty massive electric fields. But as I'll go on to say, we think these charges are pretty close, so that kind of makes sense for individual charges that are only a few nanometers apart. Okay, so to just sort of like recover what we think is going on, we think excitons are being formed, they're making, they're undergoing charge transfer, we're generating carriers. After we generate these free carriers, we see this buildup of electric charge within our device morphology. We think that's because the carriers are migrating into lower energy aggregate domains. Yet they're still being held almost at this arm's length from each other, only around 5 or 6 nanometers. But that electric field and that polaron signal is holding, it's not decaying. So we know that they're not recombining with each other. So this is really analogous to what we know happens in capacitors, where charge, electric charge builds up. And one interesting thing to add to this is that we know that for these organic materials, the dielectric constant is pretty low. So it's around 2 to 4. And while this low dielectric constant is often seen as a disadvantage for organic materials, in capacitors, what you want in the intermediate material between the metal plates is something that is really low in dielectric constant, so that you can polarize that intervening medium. And that acts as further shielding charges from mutual attraction, which are on these charged metal plates. So we really think that this must be happening at this intervening layer if you do have such large electric fields existing in this morphology. Very briefly, I'll mention that this effect is interesting. It doesn't scale the same as all the other absorption signals you see in these experiments. They're going to scale linearly, like with Beer's law, but this scales quadratically. So as we did thinner and thicker films, we saw this signal become really obvious. And we think that this is maybe a clue that it could be in other devices, maybe ones, material combinations people have looked at before, because usually with transient absorption, you don't do like super thick films for these measurements, or it becomes quite difficult. So I think this is an interesting point looking forward to other systems. Okay, so to summarize the sort of bad and the good device here, we know in the 5% device, we make free charges, but then there's a recombination pathway that I didn't talk in too much detail about through triplet and singlet CT states. This is a bit like what happens with recombination in an LED and triplet and singlet spin ratios. And then we have a loss pathway that we see form in our transient spectroscopy to triplet excitons. And that's a terminal loss pathway in this type of device. In contrast, in the device that works well, we make a bunch of free charges. We see them live on for a significant, decent period of time. And then after they're generated, we see this buildup of electric fields that resembles what we're talking about in our capacitor type system. And that's caused by the morphology of the bulk heterojunction. Okay, so having areas of charge accumulation, high charge density in these little pockets kind of seems like it would be a bad thing, kind of like a trap. But we know that our device works well. We were also able to prove that recombination, bimolecular recombination, was really low in these devices. In fact, using this metric that's known as a Langevin reduction factor, it's around 2,000 times less than would be predicted by this theory. We believe that that's the highest reported in any technologically relevant organic OPV system. So it's kind of a counterintuitive example, but we're able to really show that this idea of having large electric fields around can be a good thing rather than some sort of trap that's inhibiting device performance. The other thing I think is important to keep in mind is that we're looking over orders and orders of magnitude of timescale here. So what we saw when we looked at longer timescales is after tens of nanoseconds, we didn't see our electric field holding anymore. It was really a picosecond, nanosecond effect. And when you think of charges diffusing around, that's really a nanomilli to millisecond effect. So we're still able to see really high fill factors actually, particularly for really thick devices and decent PCEs. Okay, so I should be wrapping up second, but I just want to talk about a little bit of follow-up research here to really look in in more detail at what is really causing this effect with the morphology. There must be something going on here that's a potential energy landscape that's holding these charges at this arm's length from one another, and they can't recombine. The two kind of ideas that we have really nailed down on is this idea of having morphological cascades, for example, with three-phase morphologies. This has been around the literature for a little while now, having areas of aggregate donor and acceptor, and a slightly higher energy intermixed region that can cause a cascade of carriers away from each other, away from the interface. And we started to see some evidence for this. I'll show some more in a moment. We have the relaxation of a polaron signal here on the time scale of this charge buildup that we see. The second thing that I think is quite interesting is just thinking about this idea that, well, people were thinking about this in the 70s with organic crystals even, this idea of blend electrostatics. So if you don't have any light falling on a film, you don't have any charges, anything like that around, just by stacking different molecules in particular orientations with respect to one another, because they're anisotropic, you can have electrostatic potentials forming just from that change in electron density from stacking alone. And there's been a lot of really nice theoretical studies on this. So here's just some pentacene molecules and fullerene molecules at an interface. And you can see a really dramatic effect on the ionization potential, on the electron affinity, depending on which way you stack these molecules. So this is really of interest. It can be a little bit tough to probe, particularly in buried interfaces. The details of this, I guess if you're doing X-ray techniques and stuff, it's really hard to pick up what's happening at the interface. But yeah, the theorists certainly have been leading and continue to do some stuff on this. So we think that this could also certainly be at play. So to follow up some of these ideas, we did a few different test experiments. We looked at three-phase morphologies by just changing the donor acceptor ratio here from 5 to 95% all the way through. And just really briefly to tell you what happened is that it was really hard to get rid of this electroabsorption signal. It was there pretty much all the time. But when we went to 95% of either donor or acceptor, we saw it completely disappear. So we could really turn it on and off with morphology. We did see a bunch of excitons and charges still around, so we're still making polarons. But that really told us that this intermixed phase is important. If you have isolated molecules in a sea of your other component, then you're not going to see this effect. The second thing we looked at is to see how the time of solvent vapor annealing changed it, what was going on. When we had no solvent vapor annealing, we know we didn't see any of this effect, and at 20 seconds we did. So we went through, and you'll see as you solve vapor anneal, you get an increase in crystallinity. You can see this grow in structure of the absorption spectra. But interestingly, in the emission, you see actually a big step change. So you go from having these, what we think is more intermixed, unpure kind of crystalline regions to then a pure kind of Oswald ripened crystal. And we had a bunch of really nice X-ray characterization, GWAX done by Dr. Valerie Mitchell. And we see the step change also in our transient spectra. So if I just hold at 100 picoseconds and look at a bunch of different samples, you can see again this effect turn on as you go to longer solvent vapor annealing times. Right at this time, we see pure domains. So that's telling us we think that the domain purity, or at least something that's happened at the interface after this threshold time, is really critical to cause this buildup of charge. And then finally, I'll just tell you, we did some harsher annealing treatments at high temperatures, and we did not see this effect go away. It's still there. We know we have from the emission intensity of domains fluorescing and GWAX pi crystallite lengths. We know we're getting really big domains, 80 nanometers of BQR in our domains, really pretty crystal scattering, but we still see this electroabsorption. So that tells us that really the size of the domain for this sort of capacitor idea doesn't really matter for this. It's the interface that's doing the work. So yeah, I should really wrap up, but I just wanted to say that we kind of have an idea of that this idea of order and disorder together is maybe not such a trap. It can really have a positive impact in an interesting way on device performance. And if you're at all interested in non-completely organic materials like perovskites, there's been some work out recently that's also shown that pockets of charge accumulation in these materials, rather than really nice crystallinity, can also have a positive effect and help stop this recombination, leading to higher efficiencies. Okay, so to wrap up, we've seen this really unexpected capacitor-like charge buildup. It's caused by the bulk heterojunction nanomorphology, and we believe it plays this unforeseen role in keeping these free carriers away from each other. We know the size estimate of this type of electric field effect. It's around 500 kilovolts per centimeter. And we see that the bimolecular recombination is really significantly decreased. We think that it's really to do with what's happening right at the interface here. We think this third phase is really important, and pure domains are important, but it really doesn't matter how big they are. So this is kind of maybe guiding us towards knowing and understanding a little bit more about the real origin of this effect. Yeah, so with that, I'll finish up. I'd like to thank Greg and the Skulls group at Princeton, and now where I am right now with Ken and Trevor at University of Melbourne. So it's our Zoom group photo now, because I haven't actually spent any time with the actual group. But yeah, all these fantastic collaborators, and thank you for your attention.
Thank you very much, Kyra. That was a very interesting talk. I'd like to just call if we have any quick questions from the audience. Just remember you'll need to unmute your microphone if you'd like to ask a question. Or you can write in the chat window in Zoom as well. If anyone has any questions for Kyra.
Yeah, hi Kyra, thanks very much for that, and apologies for arriving slightly late. So if my question relates to the first few minutes of your talk, just tell me, turn up on time in future. Okay. But no, I mean, I have a question about dielectric constants. You mentioned that organics have dielectric constants of between 2 and 4, but of course the dielectric constant is frequency dependent. And so I was wondering whether you had a feel for the capacitor model, depending on the frequency that you might have, or yeah, sorry, the capacitor model based on the dielectric constant based on the frequency. So for example, if you had a static dielectric constant of, 10 and an optical dielectric constant of 4, would that change the way that you viewed your compounds, or the other way around? I was just curious.
Yeah, I'm not sure actually. Maybe you could inform me on some more details on this. I guess so. Like regardless of what's happening optically, we know at this point we have big fields that are being created by these charges that are about 5 nanometers apart, and we know that the optical absorption of molecules in the ground state, their energy levels are changing because of that field. So the molecules that are in between, if they have just any type of low dielectric constant, we believe that their electrodensity should be polarized by those big fields. So it's kind of yeah, I don't know how you would measure this and get more details, but I don't know. Do you think that there would be an effect with the frequency, or how you would probe this in more kind of detail?
Yeah, I mean, you'd have to make compounds with similar optical properties, but with different dielectric constants. I mean, making something which has got similar optical properties, but a low frequency dielectric constant which is higher, is relatively straightforward. And so that's one way you could probe the low frequency dielectric constant. I think to do the high frequency, then of course you are beginning to change the properties of the material itself. I mean, it would be interesting just to measure the optical frequency dielectric constant of the materials.
Yeah, definitely. I mean this is really the first time this effect has been reported. So yeah. I'm still trying to figure out why it's in this material and why if it's in other things. So yeah, definitely lots of things that we can do.
Yeah, no, it's as I said, it's really interesting to hear that.
Yeah, well, thank you.
Yeah, I agree. It's very interesting. I think we're already a little bit behind our schedule, so I'm really going to have to cut it off. So good on you, Paul, for jumping in and being the one to have the privilege of asking the question. Everyone, please join me in thanking Kyra for a very, very interesting talk. So our second speaker, Andrew Danos. So Kyra, do you mind that
Oh,
you
yeah.
stop sharing your screen, and then Andrew, could you